Hey guys, welcome to my channel. This is Omol the Great, and today we are here for cell physiology. Well, following my first video, I thank you so much. Guys liked it, people learned a lot, and they're asking me to shoot more videos. I promise I'll be shooting more videos. All you have to do is subscribe, share, and comment and like my video, and that's all you have to do. So welcome to my channel once again, and today I'll be taking you through cell physiology. So to start with, today on cell physiology, this is what we are going to cover. One is what is a cell, okay? Then we are going to cover cell discovery. We want to know about a brief history. Where did this aspect or concept of the cell come from? We want to know which scientists are behind the discovery of the cell. We want to know about a bit of the cell theory. How did it come about and how did it come to existence? We want to know about the cell structure and then the cellular organelles, as we shall be talking about. So to start with, what is a cell? A cell, this is a basic structural and functional unit of a living organism. So literally what this means, if you have no cell, then there is no life. Because if a cell is the structural and functional unit of a living organism, which means it's a foundation, it is a basic thing that we require to make life. So if there is no cell, there is no life. All right, so as we move on to cell discovery, there are wonderful scientists that actually came up with this concept of a cell. The first is Robert Hooke. This was around 1665 when Robert Hooke actually did some studies and observed a cell in dead cell. He observed dead cells in cork cells which are found at the back of a tree. And so he discovered and told us he was the first to crown the idea of a cell exists, okay? Then Anton van Leeuwenhoek, he was the second person to tell us more about a cell, and he was the first to observe a cell which is exactly a live cell, okay? So Robert Hooke did cell observation in dead cells, and then Anton van Leeuwenhoek did this in live cells, okay? Surprisingly, about 330 years ago, you would literally not know that there existed a cell. And not until the discovery of a microscope, that is when the concept of a cell came into existence. So these wonderful scientists played a very big role in discovery of a cell. Then Robert Brown, this also came in. He was a wonderful scientist. He was the first to discover actually what we call the nucleus. As we shall know, when we look at the structure of a cell, there are different components. Among one important component is actually the nucleus. Then there were some German botanists, as we shall see. These three that are remaining, these were all German people, okay? So these German botanists, Matthias Jakob Sheldon, he was the first to actually conclude, out of his studies, to conclude that actually cells make up plants, okay? That was around in 1838, followed by another scientist called Theodor Schwann. Theodor Schwann followed the following year in around 1839, and he told us that actually living animals actually are made up of cells. Then finally, a gentleman called Rudolf Virchow. Rudolf Virchow, he wrote this for him. He was a German physiologist. This was in around 1855, and he came up and actually concluded that actually new cells originate from pre-existing cells. Okay, so for you to have a new cell, you should have, or it should come from a cell that previously existed. And then these three actually came to explain, from their conclusions, a cell theory was developed. So the second thing we're actually looking at is what we call, the other thing is the cell theory. So cell theory, you realize they crown this cell theory, and in summary is put in this way. One, that the cell is the structural and functional unit of all living organisms, okay? Then two, all living organisms, both plants and animals, are all made up of cells. The third thing is that all cells actually arise from pre-existing cells, okay? So I think this summarizes a lot about, brief about this, what is a cell, the cell discovery, and the cell theory. And straight away, we're going to move into what you call the cell structure and want to know the different function. Our approach to this is going to be, we are going to look at the different components of the cell, and then we shall attach function to each of them. So when you come back to the cell, so literally this is a brief structure of a human cell, okay? And in these cells, literally it has primarily we can the structure of a cell is comprised of: one, we have the nucleus, okay, and then two, we have the cell membrane, which encloses the cytoplasm. Remember, the nucleus also has a fluid a fluid within called the nucleoplasm, and so it's surrounded by a membrane called the nuclear membrane. So let's start with the nucleus. So this thing here is actually called the nucleus, okay, and then right here is what we call its nuclear membrane, okay. Then the other thing, actually, so inside here we have something called a nucleolus, okay. So when we look at the function aspect of each of these, the nucleus, we know it's a control center of the cell, okay, and being a control center, it has specific important structures that actually are within that make it a control center of the cell. One, if this is to bring this out, okay, so the nucleus, it has a cell membrane, okay, but this cell membrane is actually has pores that actually linked to another structure which we call the endoplasmic reticulum. So within this we have something called a nucleolus, and then the outermost is what we call the nucleoplasm, okay. And in this nucleoplasm we actually have chromatin, okay. So chromatin, this makes part a lot of what our genetic material, which is the DNA, and then the nucleolus contains a lot of the RNA material, and you know these play a very big role in actually protein synthesis. So the cell plays a lot of protein synthesis, and it has a genetic input or the DNA input right away from the nucleus. So this nucleus, as I've already illustrated here, is in close association with what we call the endoplasmic reticulum. The endoplasmic reticulum is right here, okay? This dotted structure that is right here. Let me use black. So this structure here, this sacs, is the endoplasmic reticulum. So this endoplasmic reticulum, these are like sac-like structures that are found just outside the nuclear membrane, and these structures are in close connection to the nucleus. Okay, so they are of two types. We have what we call the rough endoplasmic reticulum, and then the smooth endoplasmic reticulum. So literally, what I've named here, this is the first one, is the rough, and this is the smooth endoplasmic reticulum. So this endoplasmic reticulum, they are among their roles. They perform a package role. So they tend to process and package content that is being synthesized and released from the nucleus. So the rough endoplasmic reticulum actually has, why is it called rough? It's also called a granular. Okay, whereas the smooth is called a granular. So which means the rough has granules, and those granules are typically the ribosomes. So why they usually stain? Because they have some DNA, they have jump genetic material, nucleotides, during protein synthesis, as we shall see. So they actually will stain. So these have the rough endoplasmic reticulum. They play a very big role in protein synthesis, whereas the smooth endoplasmic reticulum play a big role in actually lipid synthesis. Whereas the other structures here, as you can see, which are typically called the organelles, of which one we've looked at the endoplasmic reticulum. The second is actually what we call the Golgi apparatus. So the Golgi apparatus, this is the Golgi apparatus, or sometimes what we call the Golgi complex. Okay, so the Golgi apparatus, this usually is in structurally you find sometimes it has a relation to close proximity to the endoplasmic reticulum, and that's a packaging role and processing. Usually protein that is synthesized all through is usually packaged in form of vesicles, which bud off in form of secretory vesicles and bud off and take the secretion to the appropriate destination within the cell or even outside the cell. So these are one of the primary roles of the Golgi apparatus. But the Golgi apparatus also can help in packaging or modifying some polymers, especially lipid, carbohydrate, protein polymers that may be had. For example, you have chondroitin sulfate and hyaluronic acid. This can actually be packaged and processed through the Golgi apparatus, and these two actually play a very big role in the make part of the ground substance of the cell or the matrix. All right, then when we look at to finish up there, so we have some small structures that we can see here. There's a structure next to the endoplasmic reticulum. That is what we call the peroxisome. Okay, the peroxisomes. Okay, so these peroxisomes play a very wonderful role. So they play what we call a detoxification role. They usually bud off from the endoplasmic reticulum, specifically the smooth endoplasmic reticulum, and they have several enzymes. And an example of the enzymes typically they have oxidases. Okay, so they do these reactions using the oxidases. So they're able to combine this oxygen, hydrogen to form hydrogen peroxide, and in presence of catalase enzyme, they're actually able to detoxify or to break down certain molecules. For example, about a half of alcohol someone takes in is actually detoxified or broken down by these peroxisomes. The other thing is actually this other small structure that we are seeing. It's called a lysosome. A lysosome. So lysosome is structurally similar to the peroxisome because this one buds off from the Golgi apparatus. So the lysosomes play a wonderful role. They have enzymes, and their enzymes, they are quite different from those found in the peroxisomes. These enzymes are actually called hydrolases. So they perform hydrolytic reactions, and these help a very big role usually in breaking down bacteria, foreign bodies, and other materials that may be as designed by the cell. Also, there are some reactions, for example, autophagy, autolysis. These are catalyzed by the enzymes that are found within the lysosomes, and so that's one of the wonderful primary role of the lysosomes. The other we can look at, we have centriole. This plays a very big role in cell division. Okay, cell division, as we shall see. Then we have these kind of black strands you're seeing. We have microfilaments, these very small ones. Okay, microfilaments, and then we have these large ones, microtubules. And these play a very big role in forming the cytoskeleton of the cell, especially within the cytoplasm. The other thing is actually the other important organelle we can look at is this, what we call a mitochondria. Mitochondria. So this mitochondria is a wonderful and very, very important organelle within the cell. So the mitochondria plays a very wonderful role, and it's usually called the powerhouse of the cell. It generates a lot of energy. It has several enzymes. You see from its structure, it's also bilayer. It has a cell membrane. Usually it is two-layered with the inside forming some folds called cristae, and those folds have interlinked with the matrix, which has a high constitution of a lot of enzymes. Okay, and these enzymes usually play a role in generating ATP, converting so many of the molecules like glucose molecules, and use them to generate a lot of ATP that can be used for running cellular activities. So that is a very wonderful thing for us to remember about a cell. And importantly, the next thing I haven't talked about is actually what we call the cell membrane. The cell membrane. So I'm going to tell you about the cell membrane. All right. Yes, so the last bit of our cell structure we want to look at is the cell membrane. So the cell membrane plays a very wonderful role on the cell and plays a protective role. As I already showed you in the other diagram, it surrounds the cell, so protects all the contents of the cell. But also, besides protecting the contents of the cell, it also allows exchange of material between the inside of the cell and outside of the cell. So it plays that very wonderful role, and we are going to see how it does much of this structurally, how it performs most of these functions. Now, structurally, it is designed as a lipid bilayer, meaning that it has two layers of lipid. As we can see here from my illustration, you see these are different lipids. Okay, so we have a layer of lipid, as you can see, this layer of lipid, and then this other layer of lipid. So it makes a bilayer. But the structural arrangement of this lipid bilayer is in a way that actually much of the these lipids have a hydrophilic and a hydrophobic end, as we shall see. And these lipids have more of the hydrophilic ends that are exposed within the cell because of the water. So they are water loving, they're hydrophilic, whereas the other bit is exposed to the extracellular environment, which is also has a large component of water that's around the interstitium, and so it's also hydrophilic. So we expect to expose the hydrophilic ends into the water environments, and then in between here is where we have the hydrophobic tails of these lipids. Typically, these lipids that occupy the biggest composition of these lipids are actually called the phospholipids, that make up about 25% of the whole total number of lipids that are within the cell membrane. And then cholesterol also plays a very wonderful role in the cell membrane and occupies about 13%, followed by the other lipids, which makes it about 4%. And some other lipids are actually what we call as sphingolipids, that have a role to play as cell connectivity and also help have a role in nerve transmission and signaling. And then you see from the membrane composition, much of the structures are made up of lipids. So 42% of the membrane is actually lipid. 55% of it is the protein component, and carbohydrate will make about 3%. So as you can see, if you look at the structure of our lipid, which we said the most of the lipids are actually phospholipids. Structurally, we have a head, which is a hydrophilic head, and then we have the tails, as you can see from this. These are hydrophobic tails. These are water hating, whereas these are water loving. So the hydrophilic head typically has a backbone, which is one, we have this phosphate head, okay, and then it has another glycerol backbone. So these together, this complex aggregation of this molecule gives us a hydrophilic head that we're actually trying to see here. And then we have the tails. These tails are typically hydrophobic tails, as we are seeing. This can be actually made up of saturated or unsaturated fatty acids. So these are very wonderful. This is an exposed molecular structure of our phospholipid. So if you go more in detail, you are going to see that actually our cell membrane is not purely lipid, as we've said. It has some protein, but the structural distribution and arrangement of this protein, one, we have proteins that span through or move through the whole length, okay? You see the whole length of the membrane. So this protein is stretching out from here up to here, from the extracellular fluid compartment to the intracellular inside within the cell. And so it's spanning through, and so this is called an integral protein. Whereas there are proteins that actually just embedded on one side, they don't cross through, or there is no connection between both sides of the cell. And these proteins are actually called the peripheral proteins. So these peripheral proteins, and I'll try to put a table here for you that will simplify things to differentiate the two. So we've said these integral proteins actually span through the whole cell membrane of the cell, for example, like we've seen here, and there among their functions we have they act as channels or pores, as we shall see, and then they act also as carrier proteins and can also act as receptors. We're going to talk more about these integral proteins when I take you through my next video, which will be on transmembrane transport. And then the peripheral will be, they usually function as enzymes and adhesion molecules. So the other thing we need to know that I haven't named on this is actually this. This is a sort of a molecule of cholesterol. Cholesterol plays a very wonderful role in making the fluidity of the membrane. You realize this membrane of the cell is actually not rigid. So this membrane is flexible, it's fluid, and you know, this cholesterol plays that very wonderful role. It's really adaptive to the changes in temperature, and so irrespective of changes in temperature exposed to the cell membrane can actually adapt to the changes, whether it's hot or cold, and that wonderful function is attributed to the lipids, and strictly we have the cholesterol. And so the other thing that you need to know about this, during the study of the cell membrane, they described this. Singer and Nicholas, these were scientists that described this structure of the cell membrane as a fluid mosaic model. And what does this mean? This is the fluid mosaic model. Fluid implying what I've just explained, okay? And then mosaic is a mixture. So this model, explaining the model of the structure of the cell membrane, the fluid mosaic model literally would refer to these components, the mixture of phospholipids, of cholesterol, of proteins, of carbohydrates that we've talked about, and in this structure of the cell membrane, and they give the flexibility and adaptability of this cell membrane to the different functions that it performs. One thing I've not told you about is usually we have some carbohydrates, okay? These carbohydrates can actually span through the membrane or even just attach the membrane. Among the primary roles of these carbohydrates is for cell-to-cell recognition. They can actually mark, they also can actually act as markers, but also so they help to also attach cell to cell. So they are, and collectively these complexes of these carbohydrates are called the glycocalyx. Okay. Yes, so thank you so much for watching. I hope this has made cellular physiology quite very simple and understandable. And please tune in. We are going to be shooting more videos. Next, we shall talk about the transmembrane transport. And yes, all you have to do is subscribe, like, share, and comment. Thank you for watching. Love you guys.